Hello students, this is a preview for exercise 12, the lac operon. This is our last online experience, if you will, for Bio 120 Lab. And this video will help you understand what the lac operon is and help you work through the worksheet for the lac operon. And I strongly recommend having your lac operon worksheet printed out or having access to it, because I won't be showing it all the time. We'll be using a simulation. I'll be looking at the simulation. I'll be reading the directions from our exercise document telling us what to do, but I'll be answering questions on the worksheet. In fact, we should get through most of the worksheet questions in this preview video. This preview video is not polished. It's not going to be short, but it will help you work through the entire worksheet. So let's get started. I am going to quickly show you a PowerPoint that has some background information that will help you understand the lac operon. So from gene to protein, the lac operon. Now, most of you have talked about how genes are expressed. So genes are pieces of DNA that are found in the nucleus of a eukaryotic cell, and those pieces of DNA can be copied into an RNA molecule. mRNA molecules then leave the nucleus, and from there they are translated into proteins. And that process is pretty complicated, the transcription and the translation process. In addition to that, turning genes on and off is tightly regulated inside our cells, and that's really important. I'm glad that our genes are carefully regulated, because why do you need to make excess, digestive enzymes if you don't have any food to digest at the moment? So our cells are really good at figuring out what needs to be made and turns on the particular genes and makes the particular proteins we need at that moment in time, and then can turn them off. And bacteria can do the same thing. Their setup is a little bit different. So we're going to take a look at what's called the lac operon. It's found in E. coli, in bacteria, and it's kind of representative of how bacteria regulate their gene expression. How do they turn on and turn off genes? There are different mechanisms. This is one type of mechanism, and it's called an inducible system. You can turn on this set of genes. Okay, so let's take a look. This is a picture from our that you've seen already if you've read the background information, and it is complicated, right? So here we're looking at a piece of DNA, and I'm going to actually make my pen available because I like to write on it. So this thing right here is our piece of DNA that's found in bacteria. And this particular piece of DNA has 3 genes. Those genes are the lac-Z gene, the lac-Y gene, and the lac-A gene. These genes code for the building of certain proteins. These are the enzymes that are coded for by those 3 genes. Probably the one that you might hear about the most is beta-galactosidase. Okay, now these genes have to be turned on and turned off inside the cell. And they're important because these genes code for proteins, code for enzymes that are involved in digesting a sugar called lactose. Lactose is a disaccharide sugar. It's made up of 2 parts, glucose and galactose, and it needs to be digested in order for the bacteria to use the sugar to make ATP. Okay, and it needs to have these 3 enzymes that are available in order to break down lactose. If it doesn't have those, it can't break down lactose. Lactose is a good sugar for bacteria, but it's not their favorite. If glucose is around, they'll use that. Okay? So it turns out that in bacteria, clusters of genes are regulated together as units. So we can turn on or turn off this cluster of genes all at one time. We call a cluster of genes that are regulated and transcribed together an operon. And this is called the lac operon, And what happens is when it's turned on, the genes will all be transcribed into one long mRNA molecule, and that mRNA molecule is then used in translation to make all three of these enzymes. When we look at what regulates the turning on or turning off of this operon, or of this cluster of genes, it has some features similar to eukaryotes. It turns out that at the front end of this cluster of genes are the regulator pieces. So together we call the whole thing the lac operon, the three genes plus the regulator pieces, the pieces of DNA that help to regulate this, the expression of these three genes. And in that regulator region are the promoter and the operator. The promoter is the area of the DNA to which RNA polymerase, it looks like a little bit of a weird triangle here. RNA polymerase binds to the promoter, and then you can see it down here binding to that promoter, and that draws in all of the machinery for transcription, and it allows the lac operon to be expressed, to be transcribed into an mRNA molecule. There's also an operator region of this gene, of this operon. The operator also plays a role in whether or not this operon is going to be turned on. And the operator here is a piece of DNA. It's yellow in this picture. And what happens in this situation is that the operator is usually bound to a repressor protein. A repressor protein can bind to the operator. And when it's bound to the operator, the whole operon cannot be transcribed. RNA polymerase tries to get onto the promoter. When the operator is bound to a repressor, it can't. And so there's no transcription and no translation of these three genes. When the repressor is not present, then RNA polymerase can hop on when there's no repressor on the operator and can transcribe these three genes. So now the question is, what controls what controls the presence of this repressor on the operator or off the operator? And is it functional? Is it useful? And yes, it is. Here's how it works. This set of genes, remember, codes for enzymes that help break down lactose. But lactose isn't that common in the environment. It's not always around. And it's not bacteria's favorite source of energy. So here's the thing. If there's lactose in the environment, this whole operon is turned on. But when there's not lactose in the environment, the whole operon is shut down, and we don't make those enzymes. And that's because when there's no lactose present, that's what's showing in part A here, no lactose. When there's no lactose presence, then this repressor can bind to the operator and prevent transcription. And that's adaptive, right? It makes sense because if there's no lactose around, why make these three enzymes, right? But when there is lactose, and lactose is shown here by this little green circle, lactose, if there's a lot of lactose around in the environment, some of it binds to the repressor, changes the shape of the repressor, and the repressor can no longer bind to the operator. And when the repressor can't bind to the operator, guess what happens? We make these three enzymes. We transcribe the lac operon and translate it into the enzymes. And so we call this an inducible system. We can turn it on. Lactose is the molecule that can turn this system on. Without lactose, the system is shut down. The operon is shut down. It's not transcribed or translated. But with lactose, lactose binds to the repressor, prevents the repressor from binding to the operator, so we get the whole transcription and translation of these three genes. So we make the enzymes, and those enzymes can break down lactose. And bacteria are happy because they have a nice source of fuel and they have a way to break it down. Okay? That's the system. It's called an inducible system. The lac operon can be turned on in the presence of lactose. Okay? We can answer all of the questions on the worksheet, part 1. Part 1 says, well, what the lac operon? What is the lac operon and what is its purpose? Well, I gave you kind of the answer right there. In question 2, it says, briefly describe the components of the lac operon. What does the promoter do? What does the operator do? And number 3, what are the 3 genes that the lac operon is comprised of? Which of these is responsible for producing the enzyme beta-galactosidase? The LAC-Z gene produces this enzyme. Beta-galactosidase is the enzyme that actually cracks or breaks lactose. The other enzymes facilitate this process. But beta-galactosidase is that enzyme that breaks or digests lactose. Okay. Moving on to question 4. What factor must be present in the environment in order for the lac operon to be induced in E. coli cells? Well, we have to have lactose, this little green blob, right? If lactose isn't present, then we're going to have a system like up here, where the repressor binds to the operator. But with lactose, we got this system, and we get the expression of the lac operon. Part 2. Let's move on to part 2. And part 2, 3, and 4 are going to use the lac operon simulator by John Darko again. We use this guy all the time. So I'm going to click on it if I can. Well, maybe I can't. Let me just see if I can click here. Get onto this system. There we go. Okay, so here we're going to go to molecular biology, lactose, the inducible system. And we are at that site on John Darko, lac operon inducible system. Okay, now in part 2, we're going to take a look at repressors and beta-galactosidase. Remember, beta-galactosidase is the enzyme that is made when the lac operon is turned on. And that's the enzyme that breaks down lactose. All right, in this model, beta-galactosidase is reported as lactase. Lactase. These are equivalent. And so when you see the word lactase, you can think beta-galactosidase. So that's just something to know. Okay, so let's read what it says we need to do. Step number 2. We're in part 2, repressors and beta-galactosidase. I'm reading the directions from the document for exercise 12. Step 2 says the default setting is to run this as a functional repressor. The repressor's working in the E. coli cell. And we're going to set up an E. coli cell with some initial conditions. We can change things. We're going to set up, it says, to set up the initial lactose conditions is having 10 millimolarity. That's just the concentration, okay? And I don't think we change anything on this transcription rate. Let's just leave that all the same. And it says, set the initial lactose concentration to 10 millimolarity and run this simulation. And we're going to be taking a look at lactase concentration, okay? That's the same thing as beta-galactosidase concentration. And we're going to run this baby, and we'll see what happens. We're measuring here the lactase concentration over time, and we watch this thing for 100 minutes. When there's lactose present, the lactase concentration starts out at nothing, right? Because in a bacterium without lactose, it shouldn't be making any of this beta-galactosidase. But as soon as there's some lactose present, then it starts to make beta-galactosidase, and it keeps making it until, at this point, I'm guessing that lactose is gone. It made enough lactase, or beta-galactosidase, to break down all the lactose, and now the inducible system is no longer induced because there's no more lactose present in the cell, and the enzyme disappears. The operon is turned off again. Okay, then it says click a non-functional repressor and run it again. Okay, this is interesting. A non-functional repressor means that it's broken, it doesn't work. Now the repressor always binds to the operator and prevents the system from the operon from being turned on unless there's some lactose. With a non-functional repressor, it would mean that the repressor doesn't work. It never can bind to the operator. when the repressor doesn't work, we're always going to be turning on the lac operon. We're always going to be making beta-galactosidase forever, no matter how much lactose is present. Doesn't really matter. And you can see that we just keep making and making and making more of this enzyme. And it doesn't really matter what the environmental conditions are here. The repressor's broken. All right? Now, it says to take screenshots of this graph. To take a screenshot, I use a snipping tool. I just search for snipping tool, and I turn it on, and I click New, and then I can just highlight what I want to take a screenshot of. And it gives me this. I can save it somewhere. I can move it around, all right? You're going to need to do that a few times. Yeah, I don't want to deal with that right now. Okay. And then we answer the questions for part 2. Okay, so I'm going back to my worksheet, and it says part 2, repressors and beta-galactosidase. So I need to insert my screenshot, and then question 1 says, what is the relationship between the lac repressor and beta-galactosidase? The lac repressor. Well, the lac repressor, when it's functional, can turn off the operon. When there's no lactose present, the repressor binds to the operator and shuts down the production of beta-galactosidase. When there is lactose present, then this lac repressor can no longer bind to the operator, and we make beta-galactosidase, and that's a good thing because we can break down lactose then. Right? So in this graph, what we can see happening is when there's a functional repressor, beta-galactosidase enzymes go up until a certain point, probably when lactose is gone. And then we see the repressor again shutting down transcription and translation of that beta-galactosidase gene. And in a non-functional repressor, we see that beta-galactosidase is being made all the time, no matter what. The repressor isn't working. It isn't doing its job of shutting down the system when there's no lactose. Okay. Question number 2 says, in this simulation, we set the initial lactose concentration to 10 millimolar instead of 0. Why did we do this? Well, if we don't have lactose, well, let's just run that. Let's just run this system. Let's redo this. Reset everything and run it with no lactose. With no lactose, in a functional repressor world, we never make this enzyme. We don't need to. There's no lactose to break down. In a non-functional world, this would be fun to see. We keep making this enzyme because there's no regulation from the environment here because the repressor is broken. Okay, you can probably answer question number 2. Question number 3 says, when lactose, so this is not the graph that we want to look at, right? You should look at your own. Okay, there's our better graph. Okay, that's the one that we really want to look at. When lactose is present, how does removing the functional repressor, in other words, selecting the non-functional repressor, affect the amount of beta-galactosidase? Well, it goes up, right? And then number 4, when the repressor is not functional, does the cell continue to make beta-galactosidase when the lactose concentration varies from 0 to 20 micromolarity? Why is it harmful for the cell to have a non-functional? So you have a little bit of work to do. I'm going to stick this puppy up there. I want to run this again. When the repressor is not functional, does it continue to make beta-galactosidase? I'm going to say I bet it does, right? We just tried that. Let's run it again. Yeah. No matter what level of lactose we have, it's going to keep making beta-galactosidase. And you can see my runs are all on top of each other. It's not paying attention to the amount of lactose, right? It's not paying attention to it at all. It just keeps making it, etc. And that's our question number 4. You can answer that one. Now we move on to part 3, repressors and DNA. We're going to use, it says, the same simulation as in part 2. So I'm going to reset this because it's getting messy. Click Reset and click on Cellular Dynamics. So under this system, we're going to be looking at a lot of different lines. We're looking at how much lactase is present. We're looking at the repressor. Is the repressor on the DNA or not? Is there mRNA of the lac operon? In other words, is this lac operon being transcribed? If there's mRNA, it is. Oh, we're going to look at presence of glucose, maybe, and how much lactose is there. This is going to be helpful. Knowing how much lactose is around is going to help us understand when this operon is turned on and off. Okay. Then it says, set the system to the functional repressor, set the initial lactose concentration, that's the sugar. It's so easy to mess up lactase and lactose. Lactase is the enzyme. Beta-galactosidase is another name. Lactose is the sugar that we want to break down. Okay? Be very careful with that language. They're not the same, right? One's the molecule that breaks down the other. Okay. All right, run this, and let's take a look at, it just says to run it. Wow, that is exciting looking. And the result will show this. Then it says run the repressor, but leave the initial, okay, and click run. This will create a graph with 2 lines. Wait a minute. I'm reading the wrong thing. Sorry, we're down here. The cellular dynamics. I'm sorry, I was reading the wrong instructions. We're on part 3, repressors and DNA. It says set the initial lactose to 10 millimolars and hit run. We did that. Now let's look at these. You're going to have to actually, for part 3, put 2 figures. You're going to have to put 2 figures into our worksheet, just so you know. Going to have to insert both of them. Okay, this is with a functional repressor, and let's take a look at what's going on. Let's see. The repressor is on the DNA, but then it disappears again, and then it comes back on the DNA, maybe down there. Oh, right here. It goes back on the DNA. Okay. What's happening in this graph is we have added lactose, so we should turn on this inducible system. With lactose present, and that's the yellow line, we should start to break it down over time, which happens, okay? Lactose gets broken down over time because the operon is turned on. Notice that when we add lactose, suddenly the repressor, that's this red line, the repressor on the DNA disappears. The repressor can't bind to the DNA anymore, which means the mRNA is being made. The operon is turned on. We're transcribing it, and we transcribe it, and we make the enzyme lactase. That's the green line. And we continue to make the enzyme lactase until when? Why do we see this sudden drop-off? Well, look at the concentration of the sugar. The sugar, lactose, is being used up, and by 65 minutes or so, it's all used up. There's no more lactose around to bind to the repressor. When there's no more lactose around, the repressor can bind to the operator on our DNA and cause the whole system to shut down. So we don't have any more mRNA, we don't make any more beta-galactosidase, and that's a good thing. There's no more lactose to break down. So what a good system this is. Okay, now it says to take a screenshot of that and then reset it, but this time use the non-functional repressor. And we're going to kind of compare what happens. In particular, I want you to look at these red and, the purple and green lines. These are the mRNA and the lactase enzyme, all right? What happens when the repressor is broken? And, let's take a look. So reset this, hit the repressor being broken, and when the wait. We forgot to do something. We forgot to add, sorry, let's redo this. We have to add the same amount of lactose. Let's try that again. Okay, that's a little better. Okay, so in this system, the repressor is broken. It's never going to work. And we've got lactose to be broken down, and so our system is making mRNA, and it's making the lactase enzyme. The green line is the lactase enzyme. That's good. We're starting to break down lactose, but when lactose is gone, when that sugar is gone, we should stop making the enzyme. But the system is broken, and instead the system keeps transcribing the operon and keeps making the enzyme. Bummer. Okay? Now it says work through the questions on part 3, and they're pretty simple questions. number 1 says, when a functional repressor is present along with an initial lactose concentration of 10 micromolarity, what happens to beta-galactosidase between 10 and 60 minutes? Well, let's redo this because we got to see that again. Okay, what happens to the enzyme lactase between 10 and 60 minutes? Let's see, that's the green line. It's going up. It's going up. That's good because we need it to break down lactose. We need it to break down the sugar. So it increases. Question number 2. Why does the concentration of beta-galactosidase drop suddenly at about 70 minutes? Well, we know that because there's no more lactose around, right? There's no more lactose. It's all gone. This yellow line is our amount of sugar, and it's gone. Don't need it. Don't need to make the enzyme anymore. Pretty cool system. That's it. There's only 2 questions on that part, guys, and we're moving on to part 4. Okay, part 4. You're going to have to insert 3 graphs this time. I'm not going to worry about that, but you guys have to do that. And we're going to have to use a slightly different version of this model. So we're going to go back up here and do the lac operon with dioxic growth. We could do a lot more with this model. We're not going to, but we are going to change the lactose and the glucose concentration this time. And we've got different things to look at. So let's see what it says that we need to do. Click on the Cellular Dynamics tab and set the initial lactose concentration to 10 micromolarity. Ching, and click Run. Well, this is going to look a lot like what we've already done. We make sure that the repressor is functional all the time in this setup. Now, this is the same system we've seen. Take a screenshot and put it onto table 3. Now, it says reset and do it again. But pay attention. I want you to pay attention to how long it takes lactose to disappear. It takes about 65 minutes for all of the lactose we had in this cell to disappear. Remember that number, 65 minutes. Okay, we're going to reset, it says, and select the initial concentration of the sugar lactose to 10, but add some glucose. Glucose. Glucose is a simple sugar, a single or monosaccharide, and bacteria love it. They are going to use glucose before they use lactose. Okay? If there's a choice, it's like, glucose is bacteria's crack. They're going to go for it. Okay? So now it says run the system again. How long? Now we want to look at I'm kind of curious. How long did it take the cell to use up the lactose? Remember I said 65 minutes before? Now it's almost 80 minutes. It took about 80 minutes to use up that lactose because what are they using instead? Dun, 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 dun. They're using glucose. They take all the glucose and use it up right away. Okay? Now it says change the initial glucose to 15 and to 20. Let's just change it to 20. You guys can do the 15. Oh, sorry. I have to reset. Change the lactose to 10. Put the glucose on 20. Okay, so now there's a buttload of glucose. They're going to use that up first is what I'm guessing. Let's take a look. No, that's kind of interesting. They did use the glucose up, but they also quickly started using the lactose up after the glucose was gone. So it didn't take much longer to get rid of all the lactose either. But you can see that they didn't start using the lactose until after 40 minutes, after the glucose, the purple line here, has disappeared. Interesting. So it looks like the cell is using glucose first and then lactose. All right? And let's see what the questions ask. Can't remember what the questions ask. We have to move on here. It gets complicated. Okay, here are our questions. When glucose is present, what change does this cause in the breakdown of lactose? Oh, we kind of talked about that. It takes longer for the cell to start using the lactose, and it takes longer then to use it up. It happens. There's kind of a shift. Don't use it right away, because I'd rather use glucose, right? Okay. 2, increasing the glucose concentration from 5 to 20 micromolarity does what? Causes lactose to be metabolized sooner? No, remember we just saw that later. Causes the metabolizes lactose at the same time as glucose? No, that's not what's happening. Causes the metabolism of lactose to occur later, after the glucose is used up. That's what it looks like to me. And has no effect. No, I think it has an effect. And then question 2, explain why metabolizing in this way might be advantageous for the cell. Think about this in terms of evolutionary adaptations. Why is it good for the cell to have this lac operon, have this system set up like this? And why does it use the glucose first and then the lactose? I want you to think about that one. I'm going to ask you to solve that one on your own. And that should be the end of our exercise 12. So you're going to have to go in and answer those questions, but I think we've kind of walked our way through the inducible system. This is a lac operon. There are other types of operons that are not inducible but can be repressed, that are always turned on but can be turned off. The lac operon is the opposite. The lac operon is usually turned off but can be turned on. It can be induced in the presence of lactose, the sugar. So there's different systems, and bacteria have these interesting operons. Eukaryotes don't have operons, but we do have very tight regulation of our genes, of our gene expression, which genes are turned on and turned off in different cells. And that's really important because it helps our cells survive. It helps them deal with the dynamic nature of being alive, right? And that's it. I hope that this helps you a lot as you work your way through this exercise. And that's our last exercise online. For some of you, it's your last exercise. For others, you have one more coming up. That's it. Thanks.